Hello everyone. So today we will discuss a tool used for protein structure prediction, Robetta. So this is the homepage of Robetta. So before we start, we need to register here on this website in order to do our modeling. So here is a registration option. I have already registered, so I won't see that registration option on my end. So you can view it here on your right hand side. So please click on that register option and fill in the required information they ask for, like name, username, password, and mail ID. Okay. So after registration, we can proceed with inputting our protein sequence. So Robetta is a user-friendly tool so that automates protein structure prediction. So all we need is just simply input our sequence and the tool will do the rest. So if we click on this add button, you can get this page. This is the submission page for Robetta, and here we have two main section, one required section and one optional section. The required section where you provide the necessary details such as the target name and the protein sequence. Um, and in the optional section, you can customize the prediction process. So first, in the required section, we have the target name field. This is simply an identifier for your submission, such as the name of the protein or a unique ID for your project. And next is the protein sequence field. Here you can either paste the sequence directly or there is a space to upload a FASTA file. Okay. Now the next step is to submit our protein sequence. So you can go to UniProt and select a protein sequence. Here I select this insulin. So then you can search for your protein and pass the sequence into Robetta. Okay. You can copy the entry name and paste it in this target name field, and from the sequence section you can copy the sequence and just paste it here. So in the optional section we have some methods uh, which allows you to customize or fine-tune your protein structure prediction, and these settings help us select the best modeling method and incorporate additional data to improve prediction accuracy. First one is Rosetta Fold. This is a deep learning based method that uses neural networks to predict protein structures. It is fast and highly accurate and does not rely on templates, which make it suitable for proteins with unknown homologs. Okay, next we have CM. This is comparative modeling. Uh, so it builds a structure based on non-templates, and this is the best choice when homologs templates with significant sequence similarity are available. And this AB, it is ab initio modeling that predict the structure from scratch without relying on any template. It is ideal for novel proteins with no non-homologs, but it is computationally intensive and less accurate for longer sequences. And we have a predict domain option here. This option automatically splits the proteins into smaller structural units or domains for more accurate prediction, and it is useful when working with the multi-domain proteins. Okay. Then, if you already have a non-structure, you can upload that PDB file here to guide the modeling process. And alternatively, you can provide a PDB ID along with the chain information here. Then we have open constraints panel and open fragments panel here. This open constraints panel, you can input experimental constraints such as residue distances or non-interactions to refine the model. And in open fragment panel, this option allows you to upload specific fragments of the protein which can improve accuracy when working with partially known structures. So each method has its strengths and weaknesses, and choosing the right one depend on the specific requirements of your project. Ab initio will be best for novel proteins, and comparative modeling is ideal when templates are available. And Rosetta Fold is fast and accurate for most proteins. So the choice is yours. Okay. Uh, okay. Now we have paste our sequence and target name. So we can submit our sequence by clicking on the submit option. We can also fill this red box. Then click on the submit option. Okay, so our job has been submitted with this ID, and the modeling method is Rosetta Fold, and our sequence is also here. Okay, so this takes some time to process and give our model, so we can wait. If we click on the structure prediction option, we can view our results by select this Q option. Then we can view our status. If it is complete, it show complete option status, and if it is running, still running, it shows an active status. Okay. To access our results, we can click here in your username, and you can click this My Queue option. Then you can view your status page, your results page. And if we click on this job ID, you can view your models and these results. So in the header session, we have a job ID. This is the unique identifier for your prediction job, and it helps track this specific run. Then we have a status. It shows complete, indicates the prediction process has finished successfully. And this method says that our structure prediction uh, was done using Rosetta Fold only. It's a deep learning based method for protein structure prediction. Okay. Then we have our username and target name. This target name shows the name of the protein sequence you analyzed. Here I use human insulin, so here it shows INS human. Then we can view its length. This is the number of amino acids in the sequence analyzed, which is 110. And we have the date when the prediction was performed, and the expiration date. And this is a result, means the result will be available until 1st March of 2025. Then here we have the sequence information. The sequence displayed represent the amino acid sequence of the protein, and this each letter corresponds to an amino acid. Okay. Then we have the 3D structure visualization. We have 5 models here. Usually first model is preferred, and here we have a 3D visualization of predicted structure of the protein, and the structure is colored with each region representing part of the protein sequence. And the table below shows domain ID. It identifies a specific domain of the protein. In this case, it is domain ID with 647470, and this domain number indicates this protein likely has one domain. And we have a pass option. It refers to the successful passing of the domain. The word meaning of pass is analyzing or breaking down something. So in the context of protein prediction tools like Robetta, pairs indicates whether the program successfully identified and processed the domains in the given protein sequence. A value of 1 means that passing was successful, meaning Robetta could divide the protein sequence into meaningful structural or functional regions, means the domains. And if passing fails, the model might not be built correctly, as a program struggles to understand the sequence or its domains. As domains are critical functional and structural units of proteins, and this passing ensures they are recognized and modeled accurately. Then we have confidence. This is the measure of how reliable or accurate the predictor structure is. Its values ranges from 0 to 1. Here is it is 0.56, meaning a moderate confidence. It is not of a good confidence. Okay, then we have a modeling method here. The method used for prediction is Rosetta Fold, which uses AI-based techniques to predict structures. And we have model span. It indicates the region of the protein sequence included in the model. For this, it spans 1 to 110, means the entire sequence has been used for modeling. And we have length and date here. And we have an error estimate graph. This error estimate graph, it provides a visual representation of how reliable each region of the predicted protein structure is, and shows the model confidence for each residues or amino acid position in the protein sequence. In the graph here, x-axis indicates the position. It corresponds to the amino acid position in the sequence from 1 to 110 in this case, and in y-axis this indicates the estimated error or uncertainty in the position of each residue in the predicted structure. A lower values imply higher confidence and accuracy in the prediction, and higher values imply greater uncertainty in the predicted position. Here in the graph, regions with high error means the beginning of the sequence show a high error, approximately here 20 angstrom, suggesting that the structure in this region is less reliable. And here in the middle region, it has lower error. It is between 5 to 10 angstrom. It indicates that the structure is more confidently predicted than the previous one. And this could be due to some stable secondary structures like helices or sheets, or better template matches in the prediction process. And in the end of the protein sequence, it shows a slight rise in error, indicating a moderate level of uncertainty. So why the error estimate graph is important? See, it helps you pinpoint which regions of the proteins are modeled with confidence or with a low error and can be trusted for downstream analysis like molecular docking or functional studies. And these high error regions might represent any flexible or disordered areas or modeling inaccuracies. And these regions may require additional experimental validation or structural refinement. So in this way, it will guide us in further analysis. So that's all about this Robetta tool. Thank you.